Division of Chemical Sciences, IISER Kolkata. Dr. Roy is a renowned scientist in the field of structural biophysics. Dr. Roy did her PhD from IISc Bangalore and then did her postdoc from US. And then Dr. Roy started her laboratory, started her research work in IISER Kolkata. And within a few days, Dr. Roy has become a renowned structural biophysicist in India. And we are all very eager to listen to Dr. Roy's talk. And Dr. Roy mentioned to me that it will be a student-centric talk. So it will be very interesting to you all. And I'm really eager to hear Dr. Roy's presentation. So over to you, Dr. Roy.
Thank you, Professor Bharti. For a kind introduction. Let me just plug in my. Give me a second. Let me first start thanking Professor Bharti for inviting me in this workshop. And you know that here we are basically interested to learn that how using interdisciplinary approaches, I would say rather multidisciplinary approaches, we can understand or we can resolve few biological problems. So that's what the approach is bioinformatics, that's what the approach we learn from biophysics, or sometime I came from a department, chemistry, but my subject line is biophysical chemistry. So right now our focus is RNA, and today I'm going to talk about that how we investigate RNA folding and function using multi-scale modeling. So this probably will give you a different flavor which you are learning in this workshop. So let me first share a few things that what exactly motivated us to look into RNA folding in connection to their function. Now when I'm talking about that, I'm interested about RNA folding, I am particularly interested about a very particular fold that is operational. So I'm not thinking about that, okay, RNAs just can fold to whatever structure, whatever fold or architecture. I am thinking about that whether we can predict certain operational fold, okay, which is connected to my biological function. Alright, so you know, few incidents actually motivated us to look into this problems. And we all know that what it is. That was an unfortunate incident that back in December 2019, when China notified the World Health Organization about several cases of human respiratory disease, and the disease later termed as COVID-19, which turned into a pandemic condition, we all know. And the accused of the outbreak, we all know, is a virus named as coronavirus, and later it was referred as SARS-CoV-2. You know, and this particular virus, as it turned into a pandemic, it's kind of a war between human race. So one side of this war is mankind, and the other side is extremely fast virus. So in one side, extremely smart human race, and in another side, extremely fast viruses. So now, you know, you can identify many articulated reasons behind their promptness, that how they act fast. So fast in a sense that their evolution is fast. Fast in a sense that how quick they can escape the immune surveillance in our body system. But there is a key operational mechanism by which they can be very fast. And among them, one stands out is ribosomal frame shifting. It's a very specialized mechanism Well, most of you probably, I do not know how much, biology in terms of translation, or transcription, or translational process. You know probably that by which the process DNA makes RNA is transcription, and the process RNA makes protein is translation. But in virus, I do not know. However, you can raise your hand that how many of you know that how translation operates. Can you raise your hand such that I can understand your pulse?
So in virus.
Huh?
In virus.
No, in general.
Translation how operate.
How it operates. What are the key components, how it operates? Do you know how translation operates in our cellular system? Even that general, so let me tell you in a very simple manner. I thought that probably some of you know, but okay. So you know what happens in translational mechanism? So from DNA, mRNA copies our genetic code. Okay, that's the transcription. In translation, that mRNA actually, with the help of ribosome, there are two subcomponents of ribosome. One is 50S, another is 30S. Okay, so this particular segment of ribosome capture a particular segment of mRNA which has been copied from the DNA. Okay, so now what would happen in our cellular system? There are cut paste which are called translational RNA, tRNA.
Yes, ma'am.
And so this tRNA carry amino acid.
Yes.
Okay, particular fragments of amino acid. We know 20 amino acids are there. So they will tag with one amino acid. And there is kind of copy paste mechanism that which codon will be code which particular amino acid, meaning that which particular amino acid will be called, okay, that is specified by this three terms codon.
Yes, ma'am.
Okay, so now you look into that, this tRNA which is bringing, the first tRNA will bring one amino acid which is tagged with here. Okay, so this one is tagged, so this is one part and this is another part which will be carrying amino acid, and this is the codon part. And what will happen, a second amino acid will come in and then another, so it will be tagged, the elongation process will go on. Okay, so suppose that this particular chain is, the first one is formyl methionine. Okay, so every protein, know that every protein starts with methionine.
Yes, ma'am.
That methionine now will form peptide bond with another protein which will depend on this codon topology, meaning that AAC or whatever it is, what is the genetic code. Okay, then suppose this is methionine, another will be histidine, another will be alanine, tyrosine. So this way the whole chain will form until one stop codon signal comes in, it will terminate the protein. Your protein is now ready, it will function. In our cellular system, the functional molecule is protein. You are breathing, who is carrying oxygen, who is carrying some molecules, you are having food. So there is always something, some micro machine is being operated who are actually helping to transport one particular cargo from one site to another site. You know that is motor protein. So this way, who are helping you to digest? These are all protein molecules which are functional. Okay, but RNA is particularly important because RNA has the code. And probably many of you know RNA wall hypothesis, which I'm not going into that detail. But this particular mechanism, let me come to the point that why in virus it is very different. You know what? That here this ribosome will now having a sleep, meaning that when this formyl methionine already made a peptide bond, this will now exit. Okay, so one chain, so tRNA's job is over. tRNA will now take an exit. Okay, my protein chain is being made. So this figure I made very in an abstract way. I was thinking that I would just briefly tell you that what is shifting, but let me help you through that. So here in the frame shifting, actually what happens that this ribosome can scan not only one particular code, which is actually happens in higher organism, it just comes in and sit in this particular GGA, suppose in that particular code, but in virus, one particular mRNA code can scan through here and here. So this is called frame shifting. If it is one directional, then it is normal. But in viruses, it can scan this way and this way, either way. Okay, so when it is this way, then it is called -1 frame shifting, meaning that one codon can translate 2 amino acids at a time. So the speed is now double. And this is one of the key mechanisms by which we cannot recognize our higher organism is falling back or faltered, I would say, by the speed. Okay, so virus is very fast in terms of their mechanism. Now, let me show you another example. Acha, let me first give you that why particularly I am interested in a particular operational fold. Frame shifting is happening, and now I think I convinced you that this slippage is very important, and this is the behind key mechanism for their promptness. Now, you know what? In mRNA, here around the 3' end, there is a structured segment. There is a very particular RNA motif called pseudonode. This pseudonode actually can control the tension release or exertion, and then can control this speed or the rate of translation. Okay, so that's what this structured portion is very important. It's like a knob that I can control the speed. Okay, so there I am interested that how it can control, and this is a that's why it's a very targeted, therapeutic target, that whether we can somehow take control over this. So this is one of the therapeutic challenge, that if you can target this human now through therapeutic, they can take a different measure and they can control this structure. Another example, let me show you that, as I told you, this is the central dogma of our life, or rather central dogma of molecular biology, that DNA makes RNA and RNA makes protein, and protein again help DNA to transcribe. So this is like chicken egg problem, many of you know. So but when transcription will be on, or when translation will be on or switch off, this is a control mechanism. So who actually regulates that? So so in this, one process cannot just go on forever. There should be some control mechanism or regulation mechanism. So in bacteria or viruses, as now we set them as our enemy, so we have to little bit understand them strategically. So here in bacteria or viruses, there is again mRNA segment referred as riboswitches. Now riboswitches can sense cellular environment by recognizing high concentration level of metabolite or excess biosynthetic enzyme production and inhibit their excess production through a large conformational rearrangement in response to metabolite binding. I'll help you. And mostly found in virus or bacteria, and it represents promising drug target for new antibiotics. Okay. So why I am interested in a particular operational fold of RNA? Riboswitches has two particular domains. One domain is referred as aptamer, another domain is expression platform. In the juxtaposition, actually, RNA polymerase cells. I talked about translation, now I will talk about transcription. RNA polymerase, it can continue the transcription of this mRNA. But how it regulates? It regulates by this mRNA structured part. When metabolite or small ligand, suppose that you would like to control methionine production. Now when methionine is in high concentration, it will bind to the aptamer, and now aptamer will be stabilized and it will send the signal to the RNA polymerase in such a way that RNA polymerase can no longer bind here. The structure is such, the fold is such. The fold of this part of mRNA is such that this RNA polymerase can no longer bind there efficiently and can terminate the transcription. Okay. However, okay, this is off state. So when transcription is off, you can see that RNA polymerase can no longer bind here when metabolite is present. However, on the other way, when metabolite is absent, RNA polymerase comfortably bind and can continue the transcription. So this is another regulation mechanism where structure or particular fold is operational to control the biological function. Okay. So SAM1, this is a particular or toxical riboswitch example which this is the aptamer component, so which controls the transcription regulation, and this is the particular ligand which binds in the core of this particular riboswitch which regulates methionine and SAM biosynthesis, and the SAM is a derivative of methyl donor. Okay, and it actually controls the methionine production. Okay, now, so you know that in this structure, in this particular fold, you see that this is an RNA, but it's not a very classical DNA-like double helix conformation. It's a very complex. Single chain has this kind of ribbon. It's just wrapped in such a way that it is a very complex fold, and there is particular motif. Again, as I talked about pseudoknot, I will define that what is pseudoknot. Okay, but again, some particular motif has been found operational. It's very crucial motif in RNA. Okay, so this is basically pseudoknot in the secondary structural level, which looks very complicated, but let me help you simply define that a pseudoknot in RNA structure is minimally composed of two helical segments connected by a single stranded regions or loop, meaning that there will be a secondary structure which will be connected to a loop. So it's kind of a triplex kind of interaction. Okay, so this looks like a knot, but not exact knot, it's a pseudoknot. So pseudoknots are increasingly found very functional or operational motif to control the functions in viruses and bacteria. So classic example, H-type pseudoknot, which is being found operational in wheat western yellow virus. It's a plant virus. So here, this is an H-type pseudoknot. Again, complex type of pseudoknot are there in SARS-CoV-2, which is again controlling the friendship chain. So this is the RNA. Now let me ask you a very fundamental question. So this is like, I think I could convince you that in RNA why a particular fold is very, very crucial. Okay. Now, here we ask a question that RNA, if you think about in simple terms, who are undergrads here, it has a backbone, negatively charged phosphate it has, and as a side chain there are bases, ATGU, you know that in RNA, which is a little bit different than DNA, ATGC bases are there, right? That you know, all know. So now, if I ask you that you have a negatively charged phosphate, now if I wrap them, they would come close, and it has a huge repulsion, right? Negative charge would repel. Then how would RNA fold? How do they fold? Right? In this certain very nice compact architecture. So that's what the question we ask. And here you can give me certain answer that, well, RNA folds with the help of certain cations. There can be a B answer that, well, you can think of that, okay, I have a huge repulsion, but at the same time I can bring my two bases and make the hydrogen bonds between them to pair it up. I have stacking interactions, but you know what? This hydrogen bond or the stacking forces are quite weak compared to the electrostatic forces. The repulsive component is pretty high. However, if you can make a very long DNA chain, DNA is somehow still stable, but what you need is counter ion, meaning that cations. It's a negative charge. You need positive charge to neutralize that or to at least minimize the repulsion, right? So RNA folds with the help of cation because negatively charged phosphate groups of RNA are opposed to folding. And in cellular condition, we need to know in our cellular system what are the prevalent metal ion which are important for RNA folding. Okay, so you know that stability we can understand by calculating free energy. Thermodynamics, basic level of thermodynamics, you all know that stability we can identify or we can quantify rather with free energy, right? Calculations of free energy. And energy has been calculated for this different metal ion, that how it stabilizes the backbone and how it stabilizes the nucleobases. And it has been found that among different prevalent metal ion present in our cellular system, magnesium is outcompeting all of them. Magnesium stabilizes the backbone most efficiently. However, potassium, you will find that potassium, it is basically stabilizing the bases rather. So there is a preferential interaction between potassium and bases and negative charge phosphates and magnesium. Magnesium particularly important to, as it is 2+ charge, you know, as it is 2+ charge, it can bring two non-local or very long range negative charges together and stitch them. Okay, and that's why this compact fold by magnesium, rather tertiary packing by magnesium is very, very important, and it's summarized in this way that this is RNA sequence, right? So RNA sequence first with the help of potassium, and remember potassium's concentration around physiological concentration or cellular concentration around 100-200 millimolar level. However, magnesium is requirement in our cellular system is small, but they are very efficient. 1 to 2 millimolar is the concentration ballpark. Okay. So this is secondary structure formation and this is tertiary structure formation, where both potassium plus and magnesium plus found adequate for the stabilization of a compact fold. Now here let me ask you another question. Now if you think about that, well, I have in my sequence, I have 10 phosphates, negatively charged phosphate. Now if I just neutralize my sample with 10 positive charge or 5 magnesium 2+, okay, 5 into 2, would it go to this fold? If you just neutralize, you experiment on this. Okay, so this is my question to you, that if you think about this, but the answer is no. If you just think about that I have 5 negative charge containing phosphates, and if I neutralize with 5 positive charge, would it form such a nice compact RNA fold, functional fold? The answer is no. It won't. It requires excess. Okay, and there, as I am from chemistry department, my interest lies that how actually what are the requirement of ion concentration driven folding and why excess is needed. Okay, with that question we try to understand RNA ion atmosphere, but before that, as I am interested in RNA folding and ions association, of course, in RNA folding, here before we try to understand all this, we need to know a little bit fundamentals, and this is very important for you students you learn about when you are trying to understand a biophysical phenomena, two things you always ask. The time scale of the process and length scale of your system. Okay, so this is very, very fundamental. Now, and this will help you rather to identify that which methodology is suitable for your phenomena that you are investigating. Okay, so this is very important. That look, so this is where I have plotted length versus time. If I am taking into account some femtosecond, picosecond related time scale involved phenomena, then quantum mechanics will be helpful. Okay, because I need to now look into very deeper, even deeper than the atomic scale, okay, which is helping me to understand electron configuration related phenomena, which are the time scale involved phenomena of the order of femtosecond, sub-femtosecond rather. If I am looking into a molecule, let's say water, and water's different degrees of freedom like rotations, translation, bond vibration, then of the order of femtosecond to picosecond, I'm talking about the range. And there, another method is coming into the picture is all atom molecular dynamics, if you're thinking about computational phenomena, of course. Okay, if you think about experimental, I am not covering it now. I'm just covering here the computational component, because the power of computation in this era is enormous. Probably you all can identify in this workshop that we can see every level of atoms, molecules, and exceeding an experiment, because we can simultaneously see many things together. That is not possible to really, dig down to your beaker where you are experimenting on to see through what's going on at every level, right? Yeah. So that's the power of computer, that you take some input and try to understand all this. So all atom explicit solvent simulation will help you, or MD simulation you identify here will help you to understand all this atomic scale phenomena. Then if you try to understand the protein folding, RNA folding, or assembly problem, okay, those are the time scale which is of the order of, again, nanosecond, micro. So this is nanosecond, by the way. But with coarse grain you can understand all this, like assembly process, okay, assembly process where something, a bigger molecules are coming together, okay, RNA, all those bases are coming together. So this is of the order of nanosecond to microsecond, even can go up to millisecond depending on the length of your system, okay. Okay, and again, this is a regime of physics, okay, or sometimes like we also take into account global motions or aggregations. Those are of the order of second, time, or even years, okay. Those are called mesoscale, okay. So this length scale and time scale problem you can understand through this, and I have just noted that in our lab we look into all atom level and coarse grain level description, okay. What is all atom level? All atom level, all participants we take into account, not at the electronic level or quantum level. However, at the coarse grain level, coarse graining means that we take only essentials atom, but the effect of side chains are embedded, or it's called implicit, okay. So we take some essential ions explicitly, but the effect of other atoms implicitly. The effect is there, but we are not taking their complete molecular existence, okay. Now, so this is basically the methodical details, and with that we try to understand different components of RNA ion atmosphere. So our study actually identified that RNA ion atmosphere can be classified into few in this following way. Particularly for magnesium, we could identify that it is classified majorly into inner sphere ion and outer sphere ion. Inner sphere in a sense that here ion can directly interact with the RNA, and sometimes it can coordinate with more than 2 phosphates, then we call it chelated ion. Okay, a chelated ion. So this is inner sphere, monophosphate coordinated, this is chelated. And if they are screened by water, because ion always has a hydration layer, okay, it is basically layered by water, and then ion directly don't interact with your negatively charged phosphate. It is screened. Sometimes you have heard the screened word, that it is screened. So it is screened by a layer of water, or it might be multiple layer of water. But often this outer sphere ions found site specifically help stitch particular fold. Okay, so this is where we found a very distinct signal, and here we apply statistical mechanics, which is our subject regime. So in this what we have quantified is called radial distribution function, or sometimes it may call pair correlation function, where you identify that around one ith atom or molecule, how the other atoms are distributed, and this is an average picture, okay, an equilibrium picture rather we say. So here with the distinct peak, you can identify your characteristic distribution, or characteristic rather cutoff, that how proximal an ion would be, whether they are directly interacting with that proximity or screened, meaning that outer sphere ion. Okay, so this is inner sphere, this is outer sphere. Okay, that atomistic simulation is kind of either suitable to characterize efficiently, and we found the consistency that where they are found the location, particularly where inner sphere is site specifically located, we found around the same position simulated equilibrium structure. In the simulated equilibrium structure, magnesium is also located, and we could distribute them now, what are inner sphere, what are outer sphere ions. Now, two of my students, Raju Sarkar and Akhilesh, they now try to identify that what are this effect of this outer sphere and inner sphere, particularly on those on this aptamer, okay, RNA aptamer, which is regulating this transcription process. So what they could distinguish that chelated magnesiums, particularly the inner sphere, is controlling the pseudo-knot factor. This is like very thorough analysis. I am not going into the very detail, but I am just giving you the keynote that the core or site-specific magnesiums are stabilizing the pK, because in the absence of them, it is affecting the pK stabilization. And outer sphere is particularly responsible for stitching another two monolocal interactions, and in fact many other global fold in the RNA. So these details I am not talking, I am just giving you the flavor that what you can do with atomistic level. So here at the atomistic level, we cannot really think of really studying the folding, the full folding, how they really fold. Okay, so that atomistic force field, with atomistic force field, we cannot really think about to dig out. So here just we at a very elevated temperature we found that okay, this pseudo-knot, in the absence of chelated magnesiums, it is breaking, and we could find out the microscopic mechanism that two inner sphere magnesiums is actually controlling or safeguarding a very core base triple. Okay, and these three among many bases, we could underpin only 3 bases which is of potential interest for the experimentalists who would do mutagenesis or would like to take a control over this transcription process. So our role is that when we predict, we help them, because they cannot really identify, pinpoint a particular residue. Suppose they would like to mutate and would like to control, or suppose that you are doing a docking study where you would like to dock to take a control over. So there is our role basically to underpin particular targeted site, which is having a very distinguished control over the RNA fold and function. Now, another observation through literature we had that, well, this particular pseudoknot, I already gave you the idea about this beast western yellow virus, which is very dangerous for in the plant level. So there we are finding that in the literature there are lots of controversies that some are saying that magnesium is needed for the pseudoknot fold, some are saying magnesium is not required at all. So in this kind of controversies, when experimentalists are having, then our role as a computational biophysics is quite a lot, and there what we try to find out when it is considerable. Here I have noted that considerable uncertainty surrounds the role of magnesium in this RNA-PK folding and function. We try to first, again, similar way, we try to characterize the RNA ion atmosphere. We find again inconsistency that potassium preferentially interacts with the bases and sugar, while magnesium preferentially interacts with the backbone. This is the reliability we wanted to gain, because always when you do computational problem, you need to check that whether you are getting consistent result with the existing experiment, and then you go on predict, because then it will be always a plus point that what you are measuring is correct or not. Okay, will convince others as well. So one of my another students, Vaishak, he was MS at that time, now in Utah University doing PhD. So he found a very beautiful spine of hydrated ion localized in this groove of RNA. Okay, however it is a triplex. Okay, but there is a beautiful groove of ions which is localized and nicely stabilizing this particular RNA, similar to DNA. DNA also you will find that DNA, how the twist of the DNA is getting stabilized. So behind the microscopic mechanism, you will find that there is a spine of hydration. Water actually arrange themselves in such a way. Okay, there is a spine of hydration that is kind of backbone of the twist DNA. So these all we uncovered with the help of this thing. Now I talked about that what we can do with atomistic simulations. So through atomistic simulations we can only underpin those which are of the time scale of picosecond, nanosecond. Okay, picosecond, nanosecond. But folding time scale is much bigger, as I told you. Right? There we need to adopt a coarse-grained approach. But before we coarse-grain, we have to identify that what we can coarse-grain, right? So what we can coarse-grain, and that you need to really brainstorm that what will be suitable. At the same time, you have to think about that RNA folding, what we would like to understand at the atomistic level. So should we really think of coarse-graining the molecule that I'm interested? Then we thought, no, let's think about coarse-graining the ion atmosphere. In the ion atmosphere, what is our problem? The problem is we would like to understand the tertiary compact fold, right? What are important? RNA is important, magnesium is important, but the background potassium buffer, that is not explicitly helping, but that is helping at the secondary structure level. But there you have to really think about the problem that you would like to target during coarse-graining. Okay, so potassium, then we basically chalked out an analytical model. Analytical means a little bit physical chemistry oriented derivation, okay, where you generate your own Hamiltonian. Okay, so you know that you require a potential. How many of, I think I have not covered much of molecular dynamic simulations. It's not loading. How much time I have, Susmita? dynamic simulation goes in this way, that in a molecule, first you need to know that what are the force acting on it at a molecular scale. Some of you know that it is distinguished as bonded and non-bonded component. Okay, so in the bonded, it includes the first, what you need to know is little bit that how you basically construct a polymer. Because these are all polymer, right, at the end of the day. Protein, RNA, these are all polymer. So two atoms you would like to bring in together, right, by a covalent bond. And this covalent bond is called a bonded interaction. The potential of this bonded interaction is nothing but a harmonic potential, okay, because the bonds stretch and compress. So this is a harmonic potential. Similarly, now you need a third molecule. Minimally you can compose the third molecule with another potential adding on this, which is angle. Okay, angle also having a harmonic motion, right? It can do this and again become white. Okay, so this angular fluctuation we also again the similar potential, meaning harmonic potential, we can encode with. Third is fourth atom to make a chain, right? So fourth atom, what it is doing, it is making the molecule rather little bit soft. So far up to angle, you think about, I have a bond, I can only make stretching and compressing, it's very rigid. Another atom, I'm making angle, it can do this, right? So I'm making my degrees of freedom. So this is called motion, right? You are making the molecule motionally alive. So now the fourth through which you can gain more motion. So now it's dynamic. So this is the minimal dynamics we would like to encode through potential. Dihedral is also through harmonic. Okay, because it can again vibrate around a mean value. You know, harmonic potential looks like this. Okay, I could have, okay, let's keep the board. So here it fluctuates around a mean value, and you can have your potential, right, for angle, for bond, for dihedral. These are the all bonded potential. In the non-bonded potential, what you have van der Waal, okay, and you have electrostatic, okay, and sometimes you think of like, okay, a little bit segmented part is hydrogen bond. Okay, so there are geometric criteria through which you basically understand this potential, which is a function of distance, meaning that any pair of atom, how close it can be or how farthest it can be, okay, that is determined by your potential. Let's say it's a V, okay, V is a potential which is function of R. So all this potential form, meaning that harmonic one is also, you can write half K R minus R naught square, where R naught is your mean value. From mean value, how it stretch or compress, right? So this mean value, we call it an equilibrium or the closest approach. Okay. Similarly, van der Waal also has a particular potential form. Electrostatic, you know, Coulomb, right? So you add this up, this added potential will determine your potential energy of the Hamiltonian. But you know that Hamiltonian is composed of both potential energy and kinetic energy. And for making the molecule motion, kinetic energy is very, very important, right? So in molecular dynamics simulation, this V, okay, this V is a potential form. And you randomly allocate first initial velocity. Now velocity is basically related with your temperature of the system by equipartition theorem. You know that equipartition theorem by any chance, by any of you? No? That each degrees of freedom is contributed by half kT. That also we can prove. Okay. So this half kT, meaning that it gives us a relation that half mv square equals to 3 by 2 kT altogether, taking 3 directions as a degrees of freedom. Okay. So this gives me the relation that how I can maintain my temperature of a system. Let's say I would like to maintain 300 K. My kinetic energy will help us to maintain. And now how kinetic energy is related to my potential energy? Potential energy is V. You are picturizing what I am saying. I do not have the equation. I am trying to give you the best the brief part, which will be important for you. So this V, from here we would like to get a force. How potential energy and force are related? Now if force, you can write minus grad square V. Okay, so grad minus grad V, sorry. So if you take a derivative of your potential, you will get force. Now if you get force, okay, if you get force, you can apply Newton's law. What? Because force equals mass into acceleration. Can we write m into dv, velocity dt? Let's take an integration here. Okay, this part time t1 t2, okay, time t1 t2 dt, and this way my force has been taken a derivative, and it's a function of r dr, r1 to r2, integrate. Okay, after doing this integration, what you need to know, that at least at a t1 time where your molecule is located, that is a kind of a boundary condition. You need to know that where initially it is located, such that you can predict at a later time where it can go. So this is called Newtonian mechanics, and this is the fundamental concept of MD simulation. That's it. And there are advanced how you can make your algorithms. This is the algorithm of MD simulation. So now you can basically predict the future of your molecule, and then uncover a lot of things at equilibrium, whether the molecule has reached equilibrium or not, identify many properties that I'm trying to understand. Okay, because you first have to make the molecule movable, let it move, let's set the equilibrium where delta G settles or many other properties settles, okay, but it will fluctuate around the mean value. Okay, then you can identify that, okay, there is not, because your initial time, molecule's initial position we do not know where it is, right? It has to come to set an equilibrium. So that the initial part is called non-equilibrium part, and then it will settle to its equilibrium, and you measure all this equilibrium property. Okay, and there comes the role of thermodynamics, free energy calculation, etc. These are all equilibrium thermodynamics. Okay, so this is like subject-specific knowledge that you need to require. Now, with this, I lost most of my slide because equation is not supported. Well, so in coarse grain, so this is like atomistics MD simulation, but in coarse grain, what we did basically, we made our salt, which is potassium buffer, implicit. Implicit, as I told you at the very beginning with this question, that RNA is stabilized by which particular component. Okay, so I gave you certain idea that magnesium, by site-specifically interacting, it can fold the tertiary structure. But I did not give you the idea how potassium is important. I am taking implicitly, that doesn't mean that I am completely nullifying its effect. Okay, but I am strategically incorporating into the potential. Okay, its effect into the potential such that my computation becomes little bit easier. Why? You all are doing this workshop in this BIF center, and probably now you know that we are limited by computational resource. Okay, how much time scale we can simulate, that is, there we have limitation. Okay, because we do not have resource. We need a big, these are big calculations, because on several millions of atoms you have to do this integration. Right? You understand the integration I'm talking about, this integration to predict the future, right? And that you have to carry forward at least the time scale of your process to understand, right? So that is impossible. So that's why we are trying to understand this with a minimalist approach, we cannot go to that microscopic level because of this limitation, because our computational resources are limited. Otherwise, better is always that you go to the quantum level and do all this integration. The more microscopic information you can get, more is better, right, always? But the problem is our limitation, computational. That's why this coarse graining is very, very important. Okay, there we strategically make a model which is called dynamic counter ion condensation model. What is counter ion condensation? As I give you this question, let me now give you this answer, that I asked you that if I give you 10 phosphates and I would tell you that counterbalance with 10 positive charge, would it go to the pole? The answer is no. So it's a combined effect actually, where magnesium, potassium all play a role, okay, particular role. Now what's the role of this monovalent ions? Because they are at high concentration, right, 100 millimolar. What happens when ions are in a very high concentration, there is a very interesting phenomena occurs. It's called counter ion condensation phenomena. Okay, there what happens that if you think that ions are scattered in all over your beaker, it's not like that. There is a significant layer. So this is called heterogeneous layer or heterogeneous distribution of the ion layer. The first layer is very, very dense around the RNA. Okay, what happens, and this is called condensed layer. It makes a very sheet of your ion layer, and that has been distinguished in experiment, NMR. Okay, so different chemical probing experiment, they could identify this layer, dense layer of ions, where specifically we model this specific ion condensation effect, okay, by generalizing Manning counter ion condensation phenomena, where what we did that we have an array of phosphate charges, okay, and what this condensation effect does, that once any phosphate groups comes little bit closer, which is called a zero length, by which the electrostatic potential is minimized, and then condensed layer is getting dense around this region. Okay, otherwise there is an entropic effect, ion gets entropic, and this one is this enthalpic and entropic balance help us to model this Manning counter ion condensation phenomena for the high concentration, which is very, very needed for potassium. So this condensed layer is the answer which is actually required, very, very requirement for the stabilization, both secondary and tertiary structure of an RNA fold. So that you need to take into account when you are doing computation. Is just not by throwing whatever kind of number of molecule and neutralize it. No, you have to understand why nature, meaning that in our cellular system there is a specific cellular concentration requirement, right? 100 millimolar. Must be there is certain reason, right? It's not like nature just abstractly set one particular concentration. Okay. So this we did, and then we first verified this model against various experiments to gain the reliability that I could not show you. But this movie, with this movie, I can tell you that we could capture, look, a very slow groove motion. The groove is collapsing and it is again coming. So this is a very small time scale phenomena. Okay, and this is driven by condensation and combined effect of magnesium as well. Okay, so this we have analyzed, and then we could identify again that whether this concentration level of magnesium is functionally important or not, and then we found that for this wheat western neurovirus, this particular, this groove collapse is actually destroying a particular base tuple, which is very, very important important for frame shifting. Now we are connecting to the function, with the function. So this structural implications we are connecting with the function, that this base triple is very, very important, and early mutational studies have found that substitution of any of this particular A25 or C8 base abolishes frame shifting activity. So we found that this base narrowing, which we are calling narrowing, we could analyze that pre-narrowing situation and post-narrowing situation. We found that in pre-narrowing situation, this particular base is intact, triple is intact, and in the post-narrowing condition, this triple is completely abolished. So then with this, we looked into this way various complex RNA system, and we could identify various things. I could not show you that. Here. Look, here we calculated the folding free energy landscape. Okay, so this is delta G, and this is very demanding calculation. You can see now that completely the pathway you can analyze, that which secondary structure is forming first, which tertiary structure forming next, all the hierarchical route. Okay, and then you can identify what is the rate limiting step. You know that highest barrier is the rate limiting step. So in this rate limiting step, what is actually forming? 0.6. So this is basically now fraction of contact, means any pair atoms, it comes together, then we are counting them as in contact, and we are calculating fraction of contact, means we number by divided by overall number, and we could identify that what are the rate limiting event. This is the really demanding and our objective to identify, and then it is consistent. As first time computationally we could uncover this folding of RNA. We wanted to have some support, and optical tweezer experiments were there, which actually found the similar event like us for this particular SARS-CoV-2 RNA syndrome. Okay, so this is where there is a pathway, hierarchical pathway which forms first, and we found that how it is connected to frame shifting. We found that there is a magnesium which is anchoring a particular motif, pseudoknot motif, and the connection we could identify. And there again, as I said, that among many molecules, our role is to underpin particular residue which now we can target. Okay, all the drug docking, or let's say in a drug development target, it can be used. And how this particular residue, 3 residues, are taking control over this tension, exertion and release that has been shown through this movie. So this is my conclusion, that in general ions stabilize a compact fold. However, compact may not be always functionally useful, it may be detrimental as well. Okay, so but we need to explore with a particular suitable method. Okay, so we are also working on various others. I will just briefly quickly move through this, that we also working on very interesting phenomena called allostery in RNA, how it works and how it basically govern the protein binding in RNA. Okay, so I'm not going through this. I just gave you the flavor that I'm working on this kind of beautiful phenomena related works. And I know that I came for a bioinformatics in this bioinformatics center, although I'm a structural biophysics, but we are also working on connecting structure with the sequence. And we just came across this pandemic, and we soaked down a lot of sequence level data. Okay, and as our background is statistical mechanics, I thought that let's see what pattern we could find in if we do multiple sequence alignment. Okay, and interestingly, one of my students, Satyam, what he did, let me just quickly tell you, and I shared this with Shumanda. Shumanda really encouraged me as well about this work, that we did an MSA, and then what we did, that we can calculate using statistical thermodynamics, you can calculate entropy. There is a particular formula with which you can incorporate information. Okay, you have to be strategic here, which information. So we calculated mutational entropy. Okay, you can calculate mutational probability, and with this formula you can calculate entropy, and then we defined a very particular response function which is in our subject regime for any matter through which we can identify a phase transition. Okay, and that is through a response function. So this is little bit complicated term because you need to know the subject little bit. Statistical thermodynamics is the subject which is helping us across different disciplines. Okay, you learn probably in your MSc course there is a you can learn in the department statistical mechanics this course. So with which you first calculate this mutational response function, and the graph that I could not show will show you a very distinct jump with which you can distinguish your variant of concern. And here was an example that once what we could identify, let me share with you and then close. We have calculated this, and let me share a very satisfactory result that we are uncovering, that when you see the timeline, I don't know whether you can see that we are drawing a lot of data. We calculated this entropy and the mutational response function. It is basically capturing the entropy fluctuation, and you see that around April-June 2020, B1 variant was there. This is for the whole surface glycoprotein. We are probing this spike protein mainly, okay, and its genomic level. And you see that when delta emerges in India around the timeline of this March-April, okay, so it is showing a very distinct jump. So we are basically proposing that right now who doesn't have a very quantitative metric to pre-alarm a particular variant of concern, and I think that this could be this is our proposition. Okay, so this is a very new result. We published our method recently, but we are also writing this to prove that this could be a good quantitative metric that you can take into account and check that whether it's really working or not, but at least it's worked on the past variant, whatever data now patient level we have. So then based on this mutational entropy, we developed a artificial intelligence based method, and we have mutational entropy is really is the best feature that is having a most weight to our outcome. But you see that with this method, AI-based method, we are now predicting new mutations. So this could be a new generation of mutation prediction. This could be immediately important for who are doing mutagenesis experiment or any genomic level mutation. Suppose you would like to know at a disease level that which mutations probably is very missense mutations. This could be of helpful. So this is with this we are having our web server. It's almost ready. Evolve. Okay, Evolve will help you to explore a lot of genomic data, both of course at the genomic level as well as at the amino acidic level from the sequence. So this is my group. Okay, yeah, the real one also is not coming. There was another slide, but at least the acknowledgement came in. Okay. So I would like to thank DBT, her Govind Khurana Young Scientist Award grant, and the DSTS-CRB Early Career Research grant, and then DBT, where we are collaborating with Professor Anshuman Bagchi. It's a national network project, and I am also participant of this workshop. So, and thank you all for attentively listening.